via Teams or Zoom. Teams is because that's what we have. You just email me, me know. We will set up a time and day. I will set up the appointment. You will have a link to Teams that you can use, and you don't have to cross the freeway if you don't need to that way. So anyhow, any questions or concerns about the exam 2? So that was our second arc. Today we're moving on to arc 3. All right. So let me see where we're at. What do mutations tell us about gene structure? So before the break, we talked about mutations, different kinds of mutations. Now we want to use mutations as tools. And mutations are the #1 tool for doing traditional genetics. Now we do have gene sequencing and some other things that we can do nowadays. But even then, if I take a gene sequence and I do a blast search and it tells me that it's probably this enzyme, related to this enzyme or has this enzymatic function, I still need to test it to make sure that that's indeed the case. I can't just assume that just because it looks like the structure of another enzyme that it actually does that. So mutations are still a key tool of genetics. And we're going to start with talking about complementation testing. And then we're going to explain this dude Binzer's experiment. And we'll talk about how he used deletions for rapid genetic mapping. So you've guys done some 3-point mapping now. And imagine that instead of mapping 3 things, you had to map 1,000 things on 1 chromosome. Imagine how many crosses you would have to do and what a pain in the butt that would be. Well, Binzer showed a better way of doing it. All right. So let's just review here quickly. A gene is a functional unit of inheritance. Now, I think your book has a slightly different definition, but this is the definition I want you to know. A gene is a functional unit of inheritance. What does that mean? Well, a gene, you think about the coding sequence. Yeah, that's the gene. But what about the promoter? The promoter part of the gene? Absolutely. Without a promoter, you don't express the gene. What about in eukaryotes? We're going to see regulatory DNA sequences far, far away from the gene. Are those part of the gene? Absolutely. They can turn on or turn off that gene in different tissues. They affect gene expression. So genes aren't just a sequence of DNA and that's it. They're that sequence of DNA and the regulatory sequences of DNA that affect the expression of that DNA. That's all part of the gene. Within the gene, we have regulatory DNA and we have coding sequence and we have some other subunits we'll talk about. The other thing is genes don't necessarily encode proteins. Genes can encode non-coding RNAs. So let's just kind of, if I had erased this, I had it from this morning, but I erased it this morning anyhow. All right, I'm going to use purple because I think it erases better on this crappy board. Oh, we got some bicarbonate over there. All right, anyhow, I digress. So in our central dogma of molecular biology, we say that we have templated replication. That is, DNA acts as the template to make copies of DNA. We talked about semi-conservative replication, templated replication. And then we have transcription. And that's the process of using the DNA template and reading it and making a copy as RNA. In fact, the way to think about this is think of DNA as the document in the archive, and when you transcribe it, you open up the filing cabinet, you pull out that file, and then you copy that file, and that file goes back into the filing cabinet just like it was before you started, but you have a copy of it. That's transcription. You transcribed it. Okay? But transcription, well, the way Crick wrote it was it goes to RNA. I'm going to split it out a little bit, though. I'm going to say transcription can take us to mRNA. It can take us to ribosomal RNA. It can take us to tRNA, and a whole host. I'm just going to call these NC for non-coding RNAs. They can be microRNAs, small nucleolar RNAs, small nuclear RNAs, long non-coding RNAs. There's a whole host of RNAs over here that are functional, but they don't make proteins. Only one of these codes for proteins, and that's mRNA. But you can't do anything with mRNA if you don't have ribosomal RNA to build the ribosome. And even if you have rRNA and mRNA, you can't build proteins unless you have tRNA to bring in the amino acids. These other RNAs are functional RNAs and have regulatory roles or other roles, right? So a gene can encode mRNA, but it can also encode rRNA, tRNA, and any of these RNAs. So genes encode RNAs and proteins. If it encodes a protein, it goes through messenger RNA. These RNAs are all end products. There is nothing after them. They are the gene product. Okay. So just make sure you have that concept in mind. Okay. So here's our deal. We're going to look at this gene called rhodopsin. Well, the gene for rhodopsin. Rhodopsin is a protein in your eyes, in your rod cells of your eyes. At nighttime, when there's low light level, but daytime too, if there's low light levels, if I turn off all the lights in the room, shut the shades in the room, and get down to really low light levels, your vision goes from color vision to black and white vision. And you typically aren't even consciously aware that you're seeing in black and white. But that's what your low level sight is. It's just gray values. And that's your rods at work. And the way your rods work is they have this protein. Why am I blanking on rhodopsin? And it's a G-coupled protein receptor. Within rhodopsin is this little molecule of something called retinol, retinoic acid. Retinoic acid, or retinol, can absorb a single photon and it changes its shape. When it changes its shape from absorbing a single photon, it affects the shape. It transmits that to rhodopsin. Rhodopsin starts a signaling cascade, and you register having seen that single photon. Now, although your retinol can detect a single photon, it takes more than a single photon in your rods to initiate a a signaling cascade. But that is how you see. And on humans, your rhodopsin gene is on chromosome 3 somewhere. Now, of course, I didn't even catch this last night because I'm so used to saying it wrong. This is the chromosome during what? During cell division, because we have duplicates, we have sister chromatids there. Imagine that we just only have chromosome 3 without its sister there, which is how it is 99.99% of the time when the cell is not dividing. All right. But somewhere on this chromosome of 220 million base pairs resides the gene for rhodopsin, or the bulk of the gene for rhodopsin, we'll put it that way. So I want to find where rhodopsin is on this 220 million base pair long chromosome, right? So how do I do that? The other thing I want to do is, once I find this rhodopsin, I want to be able to translate, to understand it. How are the base pairs in DNA, how are they arranged to encode rhodopsin? Are they all in one block? Are they, this is supposed to show some at the top, kind of weird. I'm not sure what that was. This is supposed to show, are they sequential? Are they out of sequence? Are they part on one strand, part on another strand? Well, you probably know this by now, but those are some of the questions that you have to answer, especially in the 1950s when we've just figured out the structure of DNA. And this is a big one. How can a single gene sustain different mutations that give different phenotypes? I mean, if you break a gene, you break it, right? It should just all be one phenotype. But that's not the case. So a single gene can give me multiple phenotypes from different mutations. It's not like there's just one mutation that's broken and I get one phenotype. So how does that occur? All right, so let's talk a little more about rhodopsin. So it is a G-coupled protein receptor as 7 transmembrane alpha helices. And down here, you guys remember G-coupled proteins from your classes, hopefully. You've got some G proteins, G alpha, G gamma beta, and they are GTP binding proteins, but they are bound to GDP until this guy changes conformation and it causes the exchange of GDP for GTP, and that causes alpha to separate from beta gamma, and then you get a whole signaling cascade. But we get mutations in this rhodopsin, and that can lead to retinitis pigmentosum is one of the mutation, or I should say phenotypes, or just general vision impairment. There's also loss of night vision. There's at least 3 different phenotypes that can occur from mutations in this one receptor. Now this is what it actually looks like here, and that dark purple in the middle, well, that's retinal. And this is retinal before it's been exposed to a photon, and this is retinal after it's been exposed to a photon. Now it takes retinal a little while to go back to reset itself to the unexposed state. And you can experience this. You can go into a room, or you go out in the middle of the night, or it's really dim light, and you'll notice that after about 20 minutes of being in the dark, you can see a lot better. And what's happened is your retinal was in this state, and over that 20 minutes, a lot of it went back to this state, so you're more sensitive now because you've reset your retinal. So this is how you see. All right. So we're going to use eye pigments. Retinal was just kind of the intro of this. I would talk about humans, but we're going to look at eye pigments in Drosophila. And so we have Drosophila that have wild type eyes, and Drosophila that have white eyes, and then we have Drosophila that have sepia eyes and brown eyes, and there's all sorts of different eye color Drosophila. And one of the things we want to understand is, are the mutations that cause these different phenotypes, is that in one gene? It's just different mutations in one gene and I get different phenotypes in this case? Or are these in more than one gene? Or is it a combination of some of them are in a different gene, some of them are on the same gene? And are they in the same chromosome? So that's kind of, do these come from different alleles of one gene? Or are they multiple genes, different genes? So that's we're going to come back to that. But we're trying to discover or parse out in this is using mutations to tell us about the structure of genes, using mutations to tell us about the function of genes, and using mutations to understand the genetic code and how the genetic code functions. All right. So this is mutations as tools. We're going to use these tools to understand structure, function, and the genetic code. All right. So here are our learning objectives up front for right now. We're going to explain complementation testing. we're going to explain Binzer's rules. Binzer did a really cool complementation experiment. And we're going to discuss how he used deletions to do mapping, to do fine structure mapping and other cool things. All right. So complementation testing. Imagine this. Let me erase some of this. If I have different mutations, but the same phenotype, so different mutations that give the same phenotype. One possibility this is the homolog here. I'll put homolog on this side. The homologous chromosome over here. One possibility, oh, I wanted to move that. There we go, is that I have one gene and two different mutations. Okay. Another possibility again, this is my homolog on this side. Is that I have two genes, two completely different genes and two mutations. So I want to figure out, do I have this case or this case? Now here's the really cool thing. If I take these guys that have two different mutations, so this guy's got this mutation, this guy's got this mutation, we can do complementation testing. So here we have my maternal chromosome, and we have a defective gene over here, and this is a completely different gene. This is the R gene over here. It's completely functional. And down here on the other chromosome, this parental chromosome, this dude's, yeah, this gene is a functional gene, and that's a defective gene. Well, we can use that. Hopefully, if I put these in the right order. Okay. This is the other, okay, so this is what I just doing. I got to remember what order I put these in. This is the other case where we have, this is a defective gene, that's a functional gene, but the other mutation is in the same gene. So before, mutations in the G gene or the R gene. Here, mutation 1 is in the G gene, and so is mutation 2, but they are different mutations. And in this case, our R gene isn't affected at all. It doesn't have a mutation in it. So we can use this to build a complementation table. What I can do is I can take white and breed it with white and see if I rescue it. In this case, well, this is the same mutation, so there's no rescue. Okay. I can take garnet, and of course, if I go with garnet, it's the same mutation, there's no rescue. But look, if I take garnet and I breed it with ruby, or vermilion, or cherry, or coral, or apricot, or buff, or carnation, I get wild type. That plus means I rescue it. So here, I got white eyes. Here I got garnet eyes. Here I got ruby eyes, vermilion eyes, cherry eyes. Here I have coral eyes. Here I have apricot eyes. Here I have buff eyes. So what does this tell me? Well, we can take these and put these into complementation groups. So if I look here, it's easy for me to find 4 complementation groups right off the bat. If you have garnet, any other one of these guys will rescue you. Ghost makes it wild type. If you have vermilion, any of these guys will rescue you, makes it wild type. Ruby in the middle, vermilion, same thing, any of these guys. And if you have carnation, Well, you don't get rescued. And so we have 4 complementation groups right here, right? Which means these are 4 distinct genes. 4 distinct genes. Well, what about all the rest of this stuff up there? Well, the 5th complementation group, and there are a total of 5 complementation groups here, is a little more complicated. Because the 5th complementation group, white doesn't rescue cherry, coral, or apricot, or buff. And this is basically just the same data down here. Those don't rescue white because they're on the same chromosome, not the same gene. These are the same genes. So this is one complementation group because this is one gene. So we're looking at 5 different genes here. And look at this. This also tells us that cherry, coral, apricot, and buff are all different mutations on one gene. Garnet is just one mutation on that one gene and only gives one phenotype. Vermilion is just one mutation on another gene that just gives one phenotype. Same with cherry. But when we get here, all of these are on the same gene. So we can use a little bit of logic. You guys may be noticing this course is about more math and logic than perhaps you thought, but that's what genetics is. To separate complementation groups. And actually, that's not even the right term. But that's the term your book used, so I'm not even going to go there. I'm just going to stay with that right now. Keep it easy. So these are different genes. 5 different genes. If we take that and we make a map, maybe this will make it make a little more sense. If I have white, this is the white gene here, and there's 0.011 map units from cherry to buff. And notice they're all W buff, W coral, W apricot, W1, W cherry. All different mutations in the white gene. All bunched together because they're all in one gene. On the other hand, here's ruby, and there's just, where's ruby? Garnet. Ruby's over here. Here we go. Here's ruby, here's vermilion, here's garnet. Notice there's no, every time we put it with any of these two, if I take a garnet and I put it with ruby, I'm going to rescue it. There's garnet and ruby right there. Oh my, I'm dying. Okay. Well, we're all dying, but that's a different story. My laser pointer's dying. I got to charge it up. All right. If I use garnet, ruby and garnet together, it rescues. Vermilion and garnet together rescues. Cherry and garnet rescue. Because if I have this guy and this guy, those are my 2 guys, when I do the cross, I'm going to wind up with functional copies of each gene. So let's see how that works. This is what I was looking for. I'll come back to the other one in a minute. This is how I'm going to explain how we get complementation meaning that. So here we have our original chromosomes, mutation 1 and mutation 2. If I have a recombination event, I can either get this guy, whose phenotype doesn't change because it's got the double mutation, but I also get wild type. I can get rescue, right? That's from in the same gene. So to rescue in the same gene, I have to have recombination. Recombination is an uber rare event, especially if I'm really close together, right? So it's going to be rare that I see rescue in those. But if they are on different genes completely, well, it's going to be pretty common that if I cross these 2 guys, I'm going to get wild type because this one's going to get a function one of these, and it's going to get a function one of those. Those are the 2 things. All right. Let me go back here. All right. So I want to talk a little bit about the size of the star. Our goal here, as part of our goal, was to find out where on the chromosome that rhodopsin gene is. But genes are all different sizes. Within those 220 million base pairs are a whole bunch of different sized genes, ranging from histone encoding genes, which are around 500 base pairs, to the largest human gene we know of, which is Duchenne muscular dystrophy gene, DMD, and it's a couple million, 2 million base pairs. Now, gene names. The DMD gene is a normal gene. We all have the DMD gene. Mutations in that gene cause muscular dystrophy, Duchenne muscular dystrophy. We name the gene after the mutation. So it's just like BRCA1 and BRCA2. Everybody's got BRCA1 and BRCA2. Those are the genes. If you have variants of BRCA1 and different variants of BRCA2, that increases your risk for having breast cancer and other cancers. All right, so we've got this idea that we can use complementation testing to find out if mutations are on the same gene or different genes, and we can even use complementation testing along with recombination, these very rare events, to see if they're on the same gene. Because we're only going to get rescue very, very rarely if they're on the same gene, but it can still happen. Yeah. So if we get rescue a whole bunch, that would indicate that they are on different genes. If we don't get rescue, that indicates that they're on the same gene. But we would still expect some rescue at a very, very, very low rate because of recombination. Okay. Let me go back to what I wanted to say here. Let me think what's next. Let me see what's next. I forgot. Oh, there's Benzer. So we'll come back here. What I want to talk about next is the terminology that we use designed just to confuse you. No, it may seem like it's designed to confuse you guys. It really isn't, but it can be confusing. Notice that there's no rescue. That is, there's no complementation here. When I have white and I cross it with cherry, coral, or apricot or buff, there's no rescue. No complementation occurs. Complementation means that if I put the two of them in there, and I put, here's my white and here's my coral, well, when it's all said and done, my little dude here gets a, what do we got here? I got to do my homologs here. There we go. So my homologs here, you get, these are parental, right? So this is, say, if they're on different genes, this one's broken, but that one works, and this was broken. All right. But I wind up with wild type because I have working alleles of both genes. Complementation. Okay. On the other hand, if this is the case, or this is a single gene, that's what we got here and down here. Here's the homolog to that gene. The only alleles I can inherit are broken alleles. I can get this broken allele for mom and this broken allele for dad. There's no complementation. However, we call this, where there's no complementation, a complementation group. A complementation group are things that don't complement each other. Things that are on the same gene are complementation groups. If they complement each other, that means they're on different genes, and we don't call that a complementation group. Perfectly logical, right? I didn't invent this. So this is a complementation group because all of these are on the same gene. Each one of the other ones is a complementation group because they're not complemented by anything else. So their own group. Okay. You can't rescue them. All right. This is not my. This picture is of Binzer in the 70s. I love this picture because he's got a giant model Drosophila. But back in the 50s, Binzer did this really important experiment using complementation. And hopefully, as we go through this experiment, complementation will begin to make a little more sense. All right. So he used bacteriophage T4, or for short, we just call them phage. And phage are viruses that infect bacteria. So phage are viruses. Viruses are inert pieces of dust. They don't use energy. They don't reproduce. They don't move. They don't do anything until they are taken up by a cell. There's various ways they can be taken up by a cell. When they get taken up by a cell, their genetic information is released, and that information is used to hijack the cell. And the cell makes the viral proteins. It makes the viral DNA or viral RNA. There's all of the stuff. The cell uses energy. The cell can move. The cell is the thing that's alive. The virus is just a package with some genetic information. Doesn't use energy, doesn't give off energy, doesn't need to eat, doesn't need to drink. It just is. The cell needs to do all those things, but if a viral DNA gets in it, well, that DNA takes over the cell. So here's this T4 phage, and what do we have? We got this really kind of cool space lander thing. Those are all proteins. And inside the capsid is our chromosome, our DNA chromosome. This is a DNA virus. All right. When this guy happens to touch some of the proteins on the surface of specific bacteria, of its host bacteria, it sticks to it, and it causes this to mechanically shoot a needle into the bacteria. And that needle injects the genetic information, the DNA. That's the only thing it does. It just sits there like dust until a bacterium happens to touch a bacteria, if the bacteria is crawling along and happens to touch it, it sticks to it, that causes it to inject its DNA. Now, your book actually has it right, but this is the slide they gave me from your book, and it's wrong. Let's fix that. Bacteria aren't alive. I mean, bacteriophage are not alive. Phage do not have a life cycle. The fundamental unit of life is the cell. Cells use energy, cells move, cells do metabolism. Viruses don't do any of that. This is the lytic cycle, not the life cycle, of the phage. So what happens? Our phage happens to get touched by this bacterium, it sticks to it, causes it to mechanically inject its genetic information. The phage genetic information causes the cell to use the cell's machinery to make phage proteins, i.e. all the proteins of the capsid, to make more phage DNA. And then it uses the energy systems of the cell. Again, the cell does this, packages the bacterium, packages the phage, puts the DNA into the capsid. It assembles the capsid, does all of the cool things. And when the cell has got so full of these phage, because the phage DNA is making the cell just do that and not worry about its other functions so much, the cell bursts, it blows up, it lyses, and the cell is no more. But now, for every one of these guys, I release between 100 and 1,000 of them when a cell blows up. And most of them are never going to be run into a cell. But then some of them are, and that's another 100 to 1,000 particles. And some of those are 100 to 1,000 particles. Okay. So this is how viruses work. Now, this assay that Benzer did, he took oh man. He took Petri dishes back in the day where they're still made of glass. Let's see, where'd my purple erases better than black, so I want to use purple. And he poured nutrient agar in the very bottom. All right. And then he took his bacteria and he used 2 different strains of E. coli. B and K, I think it's B and K. So you use one strain of the other, not both at the same time. But he mixes the bacteria with his phage plus agar. And he melts the agar very carefully so it doesn't destroy the bacteria or the phage. And he pours the molten agar with the phage bacteria mixture on top of the nutrients. Now the nutrients are down here. This is going to have all sorts of bacteria all over the place and phage particles. Let's talk about what happens if the phage particles don't infect the bacteria at all. What happens is you get a lawn of E. coli. It's opalescent. It's kind of like this kind of opalescent lawn. It's very uniform. The E. coli are growing, they're happy, they're eating the nutrients. Remember, the virus doesn't care about the nutrients. All right. But if the virus infects some of the E. coli, what you wind up with, here's my nutrient agar. And on top of that, I'm going to get these regions where a phage particle infected an E. coli, and there are no living E. coli here or here or here because that phage particle burst and put out 100 to 1,000. So in this whole area, no E. coli can grow because there's these infections. And what you wind up with are these plaques. So the E. coli are actually all the surrounding area that looks kind of, I don't know what color that is. But the non-circular areas, that's the E. coli. These circular areas are where a phage particle infected an E. coli, and because that E. coli burst and put out more phage particles, that we have no E. coli growing. They're called plaques. Okay. Now, the phage, we said we had a couple different strains of E. coli, but we have a couple different strains of phage. One is called the R2+, or you can think of it as wild type. The R is for rapid. When you put these guys in with the B. E. coli, they make large plaques because this rapidly infects, so you get pretty good sized plaques. But then he's got the R2-, and that's got a mutation. And that mutation changes the host range. So think I've got a better illustration of that on the next slide, so let me go there. Oh, I got serial dilution on the next slide. Here's where I have a better illustration of it. So here's his T4 strains, his wild type, and the mutant. Here's E. coli B, here's E. coli K. When he's got the mutant and B, he gets large plaques. The mutant in K, no plaques. It doesn't infect K. K has lost its host range for the K strain. The R2 minus mutant has lost its host range for the K strain of E. coli. It cannot infect the K strain of E. coli. Maybe that E. coli doesn't have the molecule on it that makes the phage stick. For whatever reason, it can't infect the K strain. But man, does it go to town on the B strain. Big plaques. Now, if he uses the wild type, it's much smaller plaques because it's not as fast acting. It's no R. But it can infect both K and B types of E. coli. So whether it's K or B, I get small plaques if I've got wild type. Ever make sense? I thought I could do this faster. I've run out of time already. I'm a minute over. It's going to take me a while to explain this experiment, so I'm not going to try to. We will start this experiment. Remember this much. On Wednesday, we'll talk more about this experiment. On your labs for this week, by the end of the day, I will have instructions for the labs published. I was working on that and other things came up, but I'm actually getting that done. So I will have instructions for your lab for you to do some research for the lab on Thursday. If you happen to be around tomorrow, it's Goats with Go Day tomorrow. I've got some cute little tiny goats that are bottle babies.